Our presentation is on ovarian cancer, the most lethal gynecologic malignancy. So in order to start talking about ovarian cancer, it's important to understand the anatomy of the female reproductive system. The ovaries are the reproductive glands and the source of estrogen and progesterone. On the left, you can see what a normal ovary looks like, and on the right is an ovary with cancer. The eggs are produced in the ovaries, and from there, the eggs are carried through the fallopian tubes to the cavity of the uterus where the fetus develops. The serous membrane covering the uterus is known as the peritoneum. Ovarian cancer arises from three main types of cells: epithelial, germ cells, and stroma cells. The epithelial cells cover the outer layers of the ovaries, and mutations in these type of cells make up the majority of the cancer. The germ cells are the cells forming the eggs, and the stroma are the structural connective tissue. Ovarian cancer is not just one disease. Mutations can occur in various cells in the ovaries or in the related areas of the fallopian tubes and the peritoneum. The five main biological subtypes are as listed: high-grade serous, low-grade, endometrial, clear cell, and mucinous. High-grade serous is the most common, taking up around 70 to 74% of all ovarian cancers. They are broad and unstable. Low-grade serous, on the other hand, are usually diagnosed at early stages and do not have invasive potential. Endometrial composes of the smooth outer layers. Clear cell are generally large, and mutations closely resembles those seen in lung cancers. Mucinous, the last one, is the rarest form and primarily unilateral. So one of the most frequently mutated tumor suppressor gene in ovarian cancer is the SWI and SNF complex. This complex is involved in preserving the epigenetic signatures that aid in repairing damaged genes. Along with that, it also maintains the growth control, division, and maturation of cells. So the actual function of the complex is to regulate gene activity by a process known as chromatin remodeling through ATP hydrolysis. This allows them to mobilize nucleosomes, which alters DNA accessibility and regulates transcription. The SWI SNF provides crucial nucleosome rearrangement, which, as seen in the figure, does this by sliding or ejecting nearby histones. The movement of nucleosomes may either increase or reduce the accessibility of a site for a DNA binding protein such as transcription factors. The complex regulates many different cellular pathways, several of which are involved in cell differentiation as well as proliferation and migration. This all leads to success in a cell. When mutations arise that inactivates the complex, it leads to dysregulation of these pathways, thereby increasing the risk of cancer development. A loss of function would deactivate key proteins that maintain the integrity of the cell, allowing for breaks, mutations, and genome instability, which is the knife to cancer health.
Okay, so Olaparib is the targeted therapy treatment drug for ovarian cancer that we have chosen to focus on. Normally, patients who take this drug are ones who have received multiple rounds of chemotherapy and their BRCA1 and 2 proteins have been damaged or not working efficiently. It is an oral drug that inhibits nuclear enzyme poly-ADP ribose polymerase, or PARP. PARP is considered a proto-oncogene. Olaparib binds to PARP and inhibits its designated function, which is to repair single-strand DNA breaks. BRCA1 functions in both checkpoint activation and DNA repair in cell cycles. BRCA2 is a mediator of the core mechanism of homologous recombination. The hallmark is genome instability and mutations in which deactivation of key proteins such as PARP and BRCA1 influence genome integrity. When BRCA1 and 2 are damaged, the only mechanism in place that mutated cancer cells rely on to repair DNA breaks to remain alive, grow, and divide is PARP. Drugs such as Olaparib that inhibit PARP, in addition to the breakdown of BRCA1 and BRCA2, cancer cells cannot tolerate because of the double-strand DNA breaks, since mutated cancer cells cannot replicate. As you can see here in this chart, how PARP repairs the single-strand DNA breaks on the left, and that BRCA repairs the double-strand DNA breaks on the right in the green square. Olaparib inhibits PARP, and then you can see it cannot be repaired on the right, thus not being able to replicate. And long story being short, the cancer cannot mutate or grow, thus Olaparib being an effective cancer treatment. The incident rates are roughly 239,000 individuals worldwide, with roughly 21,290 in the USA. As you can see in the picture, the rates per 100,000 individuals is 11.5 for Caucasians, 10.3 for Hispanic, 9.2 for Black, 9.1 for Asian Pacific Islander, and 8.3 for American Indian and Alaskan Natives, respectively. Highest rates of ovarian cancer diagnoses are in North America and Europe, and moving from one country with low rates to a country with high rates leads to a greater personal risk. Here are multiple charts to show the rates across different geographical locations. This chart on the left shows the age-adjusted rates worldwide. You can see that Central and Eastern Europe is the highest and Micronesia is the lowest, also shown in the chart on the top right. This could be due to technology in more developed countries allowing screening to be more accessible, increasing incidence rates. The bottom right chart also shows rates across the past decade in different countries. For example, in the past decade, Japan shows a steady increase while USA steadily declines. This also could be due to environmental factors and the access to new cancer treatments, such as Olaparib. As you can see in the chart on the left, there is an increase in deaths by age group from 1990 to 2014, with the blue line being 80 plus, green being 70 to 80 years old, red being 60 to 70 years old, and the black line being 50 to 60 years old. The conclusion you can draw from this graph is that the younger the individual is at the time of diagnosis, the less likely they will die from ovarian cancer. The chart on the right shows how the percent of new cases rises with age until about 50 to 64 years old, and then it steadily declines. This could be due to the fact that older individuals are less likely to be diagnosed with ovarian cancer, and younger individuals are less likely to be screened for ovarian cancer in addition to other health factors. Lastly, this chart shows the survival rate for ovarian cancer after being diagnosed. You can see as age increases, survival rate decreases. This can be also due to multiple reasons such as risky or harsh treatments after diagnosed, or health factors such as heart disease or stroke complicating recovery times and inhibiting treatments.
So for the risk factors, we included 2 hereditary and biological reasons. The hereditary is a minority of cases, being 10 to 15% of the cases, and these include mutations in the BRCA1 and BRCA2 genes. Also mutations in DNA repair genes, such as BRIP1, RAD51C, and RAD51D. In the biological reasons, there's the incessant ovulation hypothesis, which is increased number of ovulation cycles, increased chance of spontaneous mutation, and the gonadotropion hypothesis, which increased levels in luteinizing hormone and follicle stimulating hormone found in ovarian cancer patients. So the older someone is, the more likely these mutations can happen for the biological reasons. But we're going to focus more on the hereditary BRCA1 and BRCA2 genes. Then there was an observational study, a case control study, where they collected samples of ovarian tumors, blood and specimen of women with ovarian cancer. Then they identified 3 common BRCA mutations found in Israel, and then grouped cases depending on different factors such as age, family history, etc. And then this is the first table where they collected, where they tested and who had mutations and who didn't. And they focused more on invasive throughout the rest of the study. So invasive, they test of the 779 ovarian cancer cases, 29.4% had a mutation, then 19.6 had the BRCA1 185 mutation, and so on. And this table shows mutation for age, mutation carriers higher below 60, and then stage, higher prevalence of mutations in more advanced cases, and morphology. Mucinous was very low. And then this is just comparing BRCA1 and BRCA2 in the study. And they found for age, more BRCA1 diagnosed under the age of 60 than BRCA2. And for stage, grade, and morphology, there is no significant difference between BRCA1 and BRCA2 when looking at these. And then family history. Having a family history of mutations increased chance of becoming a case. And researchers actually saw for first-degree family history of BRCA1, there was a BRCA1 mutation saw an increase in prevalence from 30% to 50%. But you can still develop ovarian cancer with no family history, as seen in a very high sporadic case, seen by the 26.5%. And the study is useful because it shows how family history can influence prevalence of ovarian cancer, and observed a 20% increase in prevalence with family history of breast and ovarian cancer, which shows a genetic role. And the study finds significant difference with BRCA1 and BRCA2 mutations in age, being more BRCA1 diagnosed under the age of 60 than BRCA2. And with this information, higher risk groups can be identified and aid in the prevention, treatment of ovarian cancer. Then this is an intervention study of the Olaparib, where they gave Olaparib plus bevacizumab, then a placebo, and the bevacizumab, and the bevacizumab targets VEGF. So 806 patients randomized, 537 were in the Olaparib and 269 in the placebo, and they looked at progression-free survival. And this graph shows that Olaparib was more effective than placebo being around 17 months, and then the Olaparib being around 22 months. But when you they separate it out, and then by BRCA1 mutations, and there was a significant difference when compared to no mutation. It increased from 24 or 22% in the placebo to around 37 with Olaparib. And then for the disease progression or death, Olaparib significantly reduced pretty much all cases such as age, stage, and then the side effects. Olaparib did reduce disease and progression, but it was followed by side effects in the all grade. It increased pretty much all grade. And then for the grade 3, there's an increase in anemia and lipemia. Then the risk-benefit analysis, Olaparib showed a significant difference in progression-free survival. Women 65 and older showed a slightly better response when compared to the placebo group. And the same thing was seen with the BRCA1 and BRCA2 mutations. But then higher percentage of side effects in Olaparib. Only big difference between Olaparib in terms of grade 3 side effects was anemia. All grade side effects higher in Olaparib group. Then the cost-benefit analysis. For Olaparib given with the bevacizumab, it costs about $6,000 per progression-free survival as of 2020. And the class of Olaparib is about $7,000 for 60 grams of tablets. And you can see that it is effective against BRCA1 mutations. And then this shows there are other treatments that are cheaper and could see the same progression-free survival.
Okay, so there are many different subtypes of the disease, the most common being serous, endometrioid clear cell, mucinous carcinoma, and the less common being the transitional cell tumors. The transitional cell tumors include the transitional cell carcinoma and malignant Brenner tumor. The subtypes used to be grouped together under the umbrella category of epithelial ovarian tumors. However, they are now separate subtypes with various behaviors. The prognosis or the likely outcome depends on the various stages. In stage 1, the cancer is found in one or both ovaries but has not yet spread. The likely outcome is great with 90% of patients living 5 years. Stage 2 entails that the cancer is now in at least one ovary and has spread into at least one other area of the pelvis. This stage has a lower prognosis of a 5-year survival rate of 70%. Stage 3 ovarian cancer is classified by cancer in one or both ovaries that has spread outside of the area of the pelvis to the abdomen and/or in nearby lymph nodes. This stage has an even lower prognosis with a 5-year survival rate of 39%. Stage 4 ovarian cancer has cancer in one or both ovaries that has now spread outside of the abdominal areas and into other places such as the lungs. The 5-year survival rate for this diagnosis is now lowered to a mere 17%. The patient experience. So in the beginning, patients may not have symptoms that they're able to notice. Many patients, they don't stand out because they're like symptoms that are common in other less serious diseases as well, such as weight loss, pain in the pelvic area, fatigue, back pain, constipation, frequent urination, and abdominal bloating. So in regards to resistance and drug combinations, chemotherapy is the most effective upon diagnosis. However, over time, cancer cases will reoccur 70 to 90% often due to resistance. Patients eventually become resistant to PARP inhibitors. Drug combinations, the radiotherapy and chemotherapy have great short-term effects and shrink tumors quickly. However, over time, can have recurrent effects. Due to resistance, this is often paired with various PARP inhibitors such as Olaparib to enhance the effectiveness of radiotherapy and initial chemotherapy. Combining Olaparib with cisplatin can be more helpful to tumors in vivo than Olaparib alone. Subgroups of varying benefit. There are different incidences by location. So Europe and America are going to have the highest incidence, with Africa having the lowest. But we don't know if this is due to lack of screening and healthcare, or if this is just actually there are less ovarian cancer cases in Europe and America. Also, in regards to age, the most common age for ovarian cancer is 55 to 64%, with 45 to 54 and 65 to 74 close behind. So rationale and development of the targeted. Olaparib and other PARP inhibitors target mostly cancer cells and avoid normal healthy cells. This works by blocking PARP inhibitors such as PARP1, PARP2, and PARP3. The PARP enzymes repair any DNA that is damaged in cells. The inhibitors such as Olaparib keep cancer cells from using the PARP enzymes to repair, resulting in death of cancer cells. However, this can also result in off-target effects where not only the cancer cells are affected. So these off-target effects that I was talking about are the effects of a drug binding to targets that it was not intended to bind to. This can lead to negative side effects. Olaparib does not only 100% affect cancer cells, but normal cells can also be affected and unable to repair damaged DNA, which results in death. Olaparib reduces the replicative S phase in certain cells, thus decreasing homology-directed repair activity. Olaparib can lead to replicative stress in p53 wild-type and immune cells. The therapeutic index, or the window between the effective dose and the toxic dose, which is based on a population level, not on an individual person. So the window is going to be where the least amount of people have a toxic effect while still having a positive effect from the drug. So for this reason, this window is often going to be narrow, with the effective dose not being much lower than the toxic dose of the drug. Olaparib is available in 100 milligram and 150 milligram tablets. The recommended dose is 300 milligrams daily, or 2 150 milligram tablets. In order to, if you're having too many symptoms from the drug, or you're not it's not effective enough, the 100 milligram tablet is available to go higher or lower than that 300 milligrams. And the following slides are our references that we used for this presentation.
Thank you.